Good afternoon, everybody, and welcome to today's webinar. My name is Abby Bronson, and I'm the SVP of Research Strategy at PPMD. And we are really lucky to have NS Pharma here today on the phone, and so appreciative that they could give some time to our community. They're going to provide us with an overview on their investigational product, Viltolarsen, for exon 53 skipping in Duchenne. I think as many of you know, they've got their early phase studies 1, 2 are completed. And they have an upcoming phase 3, and we'll hear all about this and where they are in the regulatory process. They've been doing really a lot of great work for our Duchenne community, and again, just want to thank them for taking the time to speak with us today. So before I introduce our speakers, I want to let everybody listening know that we'll have time for questions at the end of the talk. So if you have a question or something doesn't make sense, just type your question in the chat box, and we're looking at those, and we'll answer as many as we have time for. And then we'll also answer any that we don't get to by email after the webinar is done. The webinar is being recorded, so we post it on our website, and that's where you'll find any questions that didn't get answered. So, and now it's my pleasure to introduce the two speakers we have from NS Pharma today. The first is Angela Melia. She's the Associate Director for Research and Development at NS Pharma. And we also have Dr. Paula Clemens, who is the Study Chair for the NS Pharma Studies 201 and 202. She's also the Medical Director for Synergy. She's a professor and Vice Chair of Neurology at the University of Pittsburgh School of Medicine, and a division chief of the neurology service at the VA Pittsburgh Healthcare System. So really want to thank these two speakers for being with us today. And now I'll turn it over to Angela.
Thank you, Abby. On behalf of Nippon Shinyaku, our parent company in Kyoto, Japan, and NS Pharma, our U.S. subsidiary here in Paramus, New Jersey, we would like to thank the Duchenne muscular dystrophy community and the Parent Project for Muscular Dystrophy for inviting us to share our corporate profile with you today and a little bit more about our Viltolarsen program. I am an employee of NS Pharma, as I just mentioned a moment ago, and the global study leader for the Viltolarsen Phase 3 program. Dr. Paula Clemens, also here with us today, is an employee of the University of Pittsburgh and the Veterans Affairs Medical Center. The University of Pittsburgh received a grant from NS Pharma to support Dr. Clemens' advisory efforts for the NS Pharma Viltolarsen program. While NS Pharma is not a publicly traded company here in the United States, our parent company, Nippon Shinyaku, is publicly traded in Japan, and therefore the content on this slide acknowledges that we will be making forward-looking statements in this presentation. NS Pharma, as we mentioned a moment ago, is the U.S. subsidiary of our parent company, Nippon Shinyaku. NS Pharma is the sponsor of the North American Phase 2 and the global Phase 3 trials of Viltolarsen. Our focus area is orphan rare disease. Our parent company, Nippon Shinyaku, is a middle-sized pharmaceutical company in Kyoto, Japan. We recently celebrated our 100-year anniversary on October 2nd. Nippon Shinyaku focuses on 4 major disease areas: rare disease, urological disease, hematological disease, as well as gynecological disease. Today we will be discussing Viltolarsen. Viltolarsen is an antisense oligonucleotide with the morpholine backbone. The morpholine refers to the 6-membered ring you see in the chemical structure on this slide. Morpholino refers to chemicals that contain the 6-membered ring. These chemicals are sometimes referred to as PMOs, especially in the medical literature. Viltolarsen was jointly invented by our headquarters, Nippon Shinyaku, with the National Center of Neurology and Psychiatry in Japan. It is an exon 53 skipping investigational drug whose route of administration is by IV infusion once per week. Here I am pleased to share with you our regulatory update. In the United States, we were granted Fast Track designation on October 25th, 2016, and granted Orphan Drug designation on January 24th of the following year in 2017. Very shortly thereafter, we were granted Rare Pediatric Disease designation on January 24th, 2017. This year, we initiated our rolling submission of the NDA to the FDA in February, and we completed the submission on September 30th. In Japan, we were granted Sakigake designation, which is a pioneer review designation system, on October 27th, 2015. And on October 22nd of this year, 2019, we were granted Orphan Drug designation. We submitted our NDA to the Ministry of Health, Labor, and Welfare in Japan on September 26th of this year, and we were granted an NDA review under a conditional early approval system by the MHLW on November 1st, just a couple of short weeks ago.
Can you just back up? Can you just let everyone know what MHLW is, because that's probably an acronym they haven't heard.
The Ministry of Health, Labor, and Welfare?
So it's the Japan equivalent of the FDA, correct?
Oh, yes. Yes.
Okay.
Thank you.
Perfect. Thanks.
Okay. Here you see a schematic drawing of a portion of the dystrophin gene. As you may know, the dystrophin gene is the longest gene in the human body, made up of 79 exons. The exons are portions of a gene linked together that provide instructions for making a protein. In this case, the exons contain instructions to make dystrophin, which is a protein muscles need to work properly. In Duchenne muscular dystrophy, there are errors in the dystrophin gene. Most often, these errors are missing exons or deletions of the exons. These create errors in the instructions for making dystrophin, and without dystrophin, muscles become weak, and over time, the muscles become damaged. Exon skipping is a potential therapy being developed for patients with DMD. Specialized molecules are created to skip over the non-working part of the dystrophin gene, allowing the remaining exons to fit together and attach. Thereby, it creates a dystrophin protein that is shorter than normal, but it is still partially functional. As I mentioned a little earlier, Viltolarsen is an exon-skipping investigational drug, and it is currently being tested in our Phase 2 open-label extension study and our Phase 3 efficacy and safety study in the treatment of boys with Duchenne muscular dystrophy who have specific changes in the dystrophin gene that may be helped with skipping of exon 53. I'd like to share with you now our Viltolarsen clinical program. As you can see, our Phase 1 program was conducted in Japan. The National Center of Neurology and Psychiatry conducted an investigator-initiated study, and our headquarters, Nippon Shinyaku, conducted our Phase 1, Phase 2 dose-finding study. Our Phase 2 program was conducted by NS Pharma in both the United States and Canada, and our Phase 2 extension study is currently ongoing also in North America. I would like to now share with you a little about our Phase 3 program. RACER 53 is our global Phase 3 study in 16 countries and over 40 clinical trial centers worldwide. At the present time, the United States and Japan are open for recruiting. The other countries will begin recruiting on a rolling basis as they each receive their regulatory approval of the study. Here we see all the 16 countries participating in RACER 53. In North America, we have the U.S. and Canada. South America is represented by Chile. In Europe, we have the Netherlands, Sweden, Russia, Italy, Spain, United Kingdom, Belgium, and Turkey participating. And in the Asia-Pacific region, we have Japan, South Korea, Taiwan, New Zealand, and Australia. A little bit more about the RACER 53 study is that we plan to enroll approximately 74 patients globally in the 16 countries. The patients will be randomized at a ratio of 1:1, that is, Viltolarsen 80 milligrams per kilogram per week to placebo. The route of administration is IV infusion, administered over a 60-minute time frame for 48 weeks of treatment. This is a schematic diagram of the study design. The screening period is 21 days in length, which will be followed by a 7-day pre-treatment window, during which a pre-infusion visit will occur. Randomization and first dose will occur on day 1, followed by 48 weeks of treatment. A follow-up telephone call will be made to our patients within 30 days of the last dose of study drug. The primary objective of RACER 53 is, as you see here, to compare the efficacy of Viltolarsen administered IV, intravenously, at weekly doses of 80 milligrams per kilogram over a 48-week treatment period versus a placebo control in boys that are ambulant from 4 to less than 8 years of age that have Duchenne muscular dystrophy mutations amenable to exon 53 skipping. And we will be using the time to stand as a measure of strength and function. The primary endpoint is the change in the time to stand from baseline at the beginning of the study to the end after 48 weeks of treatment. The hypothesis is that an improvement in function is due to an increase in muscle tissue dystrophin expression. We are also exploring secondary endpoints in the RACER 53 study. These secondary endpoints are time to run walk 10 meters, the 6-minute walk test, the North Star Ambulatory Assessment, also known as the NSAA, and the time to climb 4 stairs. Quantitative muscle strength will be measured by a handheld dynamometer. We will be measuring muscle strength on the dominant side only for elbow extension and flexion and knee extension and flexion. As in any clinical trial, there are key inclusion and exclusion criteria. Some of the key inclusion criteria of RACER 53 are: the boys must have a confirmed diagnosis of DMD as defined as male with clinical signs compatible with Duchenne muscular dystrophy, confirmed DMD mutation in the dystrophin gene that is amenable to skipping of exon 53 to restore the dystrophin messenger RNA reading frame. The age, as I noted earlier, is 4 years of age to less than 8 years at the time of the first dose. Able to walk independently, and that means without any assistive devices. Able to complete the time to stand without assistance in less than 10 seconds. And they must be on a stable dose of glucocorticoid for at least 3 months prior to the study entry. Some of our key exclusion criteria: current or history of chronic systemic fungal or viral infections, positive for hepatitis B antigen, hepatitis C antibody, or HIV antibody at screening, any previous treatment with Viltolarsen, if you're currently taking an exon skipping agent or have taken any other exon skipping agent within the 3 months prior to the first dose, or previous treatment with any gene therapy will be exclusionary. I'd like to get a little bit more into which function and strength tests we will be doing. As I noted before, the time to stand test, it measures the time required for a participant to go from lying flat on the floor to standing. It will be administered independently and as part of the North Star Ambulatory Assessment. The time to run and walk 10 meters measures the time for a participant to walk or run 10 meters, including a 6-point rating scale for the quality of how they run and walk. Again, this is administered independently and as part of the NSAA. The North Star Ambulatory Assessment, or NSAA, is a clinician-rated 17-item functional scale. The NSAA assesses functional activities including standing, getting up from the floor, negotiating steps, hopping, and running. The test will take approximately 20 minutes. The 6-minute walk test is considered a simple, standardized, and low-technology, cost-effective means of clinically assessing functional motor status, as well as integrated global response to exercise. This test will take approximately 30 minutes to complete. The time to climb 4 stairs, as the name implies, assesses the time it takes a study participant to climb 4 stairs. The test will take approximately 5 minutes. The steps used in this examination are standardized in their tread width and height of the rise. Muscle strength will be measured as well, as I noted, for elbow extension and flexion and knee extension and flexion, on the dominant side only using a handheld dynamometer. This test takes approximately 15 minutes. A Racer 53 informational website will be available in the United States and in select countries. The website will provide study-specific information to multiple audiences, including caregivers, as well as referring physicians. The study website will include information about Duchenne muscular dystrophy, an overview of the Racer 53 study, a clinical research overview, as well as list our Racer 53 clinical trial site location. Additionally, we plan to include in this website a brief pre-screener for families to be able to see if their child may qualify for the Racer 53 study. To promote study awareness, online advertisements regarding Racer 53 will be placed on social media sites such as Google, Facebook, and Instagram. It should be helpful to direct any interested patients to the study website where they can gather more details. Qualifying patients will have information collected in a notification email sent to the nearest clinical trial site to where the patient lives. A HIPAA-compliant portal will house the referral contact information, and this portal is planned to be updated throughout the pre-screening process. The Racer 53 Phase 3 study is posted on clinicaltrials.gov, and you can see our clinicaltrials.gov identifying number is NCT 04060199. Alternatively, more information about the Viltolarsen Phase 3 program is available on our NS Pharma company website. Just click on our pipeline and then click on the clinical trials link. On behalf of NS Pharma, Nippon Shinyaku, and the Parent Project for Muscular Dystrophy, we thank you again for inviting us to share an update on our Viltolarsen program. And here you can see a thank you in all the different languages for the countries participating in Racer 53. At this point, I'd like to turn the session back over to Abby of PPMD to take any questions you may have for us. Thank you very much.
Thank you so much. That was really informative, and looks like it's really a promising program. We're so excited. So one question, I think that, I think all of us are very used to these exon dystrophin restoration programs is, along with the outcome measures that you're collecting is to collect biopsy data to measure to quantify the dystrophin. And I didn't hear you mention that. Can you talk a little bit about that?
We have conducted muscle biopsies in our phase 2 program, so the decision was made that we wouldn't be collecting it in phase 3. But we do have that data from our phase 2 program.
Great. Okay. Thank you. Let's see. I think there have been some questions that came in previously, and a lot of them are sort of regulatory related, your regulatory process. So just to go back, and you probably mentioned some of this in your talk, but so has the NDA been accepted yet here in the U.S.? What's the process for that?
Sure. Yeah, we completed our rolling NDA submission to the FDA on September 30th. We're now waiting to hear back from the FDA on whether the NDA submission is complete. And at that point, the agency will begin their comprehensive review of the full NDA submission. We hope to provide updates on the status of our NDA to PPMD and the Duchenne muscular dystrophy community in the near future.
And is there a timeline? I mean, the FDA is always so rules down and has times for everything. Do they give you a length of time that it will take them to determine whether your NDA submission is complete, or they just can do it whenever?
I'm not able to really speculate on the time frame for for hearing back right now.
Okay.
Thank you.
And the goal is accelerated approval. Is that correct?
Yes. The FDA's accelerated approval pathway is designed for the FDA to review new drug applications and submissions for certain diagnoses and conditions based on what they call a surrogate endpoint. And this may include a laboratory measurement. In the case of our Viltolarsen program, that surrogate endpoint would be the change in the dystrophin expression as shown by Western blot. So yes, the goal is hopefully accelerated approval.
Okay. And then you have this confirmatory study ongoing, so that's great. And then in Japan, so you mentioned that you had was accepted under some conditional approval scheme. Can you talk a little bit about that?
Yeah, our parent company in Japan submitted our NDA to the Japanese Ministry of Health, Labor, and Welfare on the 26th, and we received an official letter of designation for their conditional early approval on November 1st. We hope to provide further updates on that, on the status of the NDA in Japan as more information becomes available.
Okay. Let's see. So one thing that I saw in the presentation, and I think that this has come before, is that so in your phase 2, you had a 40 mg dose and an 80 mg dose, and then, but you're going forward in the confirmatory, the Racer 53, with an 80 mg dose, and everybody in the phase 2 are on the open label extension on their dose, their original dose. So is there any plans to eventually move them up to the 80, or can you talk a little bit about that?
Yeah, thank you. We appreciate the concerns regarding this issue. Research studies are conducted to obtain the best possible information of whether a new drug will be beneficial for the treatment of patients. There is a tremendous appreciation of those families who are participating in research, which has the ultimate goal of providing conclusions based on the human clinical research for the patient community. And our decision of what dose to test in the 301 study was based on an in-depth view of the safety and efficacy data that we have obtained thus far in the open label studies, together with guidance from the regulatory agencies. So based on the discussions, we reached the conclusion that the approach that was most likely to produce a clear and confirmatory data for the new phase 3 study was to test the 80 milligrams per kilogram against the placebo in this randomized, blinded, controlled fashion.
Okay. So the boys that are in the open label, that are on the 40 will not be moved up to the 80. I mean, I guess it's, back to the potential of direct benefit. And so if you're saying that 80 is the dose, I mean, I know there's trade-offs to everything. So we're just wondering if those boys in the open label extension who are on the 40 mg will ever get to the 80 mg dose.
Dr. Clemens, would you like to add anything further to the question?
Sure, and just to let people know that I am the study chair on the phase 2 study, so been very involved in this since the beginning of the 201 study and continuing with the 202 study. So just as a reminder to everyone, at the point that the phase 2 studies were started, the highest dose that had been given was 20 milligrams per kilogram in the phase 1 study, and there was a very detailed examination of the preclinical data and in discussion with the FDA to determine what were the doses that would provide an acceptable risk to test in the phase 2 studies. And from that, the decision was to test both 40 milligrams per kilogram and 80 milligrams per kilogram. And so 8 participants were enrolled in each of those dose categories in the North American study, and those 16 participants continue on drug at this time. It's really important to remember that we're in a reasonably early phase in drug development. I know for the families that are involved, some of them have been in the study for 2 to 3 years, so it's easy to sort of lose sight of the fact that this is really research, and we are continuously looking at the safety and efficacy of the drug dosage. And really, we all have the same goal, which is to find out whether this drug will be beneficial for those Duchenne patients who have deletions that are amenable to exon 53 skipping. And as a study team, we feel that we can best get to that goal by continuing both of the dose levels in the phase 2 study. It will provide the best ongoing data to support the drug approval process, and has already provided very important data to inform the design of the phase 3 study. So it's a little bit of a complicated answer, but it's a complicated question, and we totally appreciate what folks are asking.
No, and that makes sense. That makes sense. Also, it was, you're going for AA, you're submitting on a rolling submission, so it's a very complex situation. So thanks for that. So now we're switching to clinical sites. So someone asked if there on the slide where you showed the number of sites, there was only one U.S. site listed. Is that true? And do you anticipate adding more sites in the United States?
There is not only one site in the United States listed. I think maybe the slide was a little misconstrued. That just showed the countries.
Okay, all right. So how many, can you remember off the top of your head how many sites there are in the U.S.?
We're still in startup. Right now there's approximately 9 or 10 that are already ready to get going with their submissions. So we do have a good representation throughout the United States, the different geographical regions of the U.S.
Okay. So let's see. So something came in about some of the exclusion factors and I think you mentioned stable dose of glucocorticoids, but are growth factors an exclusion item?
So this is Dr. Clemens. I can address that. I think perhaps the question is regarding growth hormone. Is that correct?
Yeah.
Okay. It is the desire of the study not to have growth hormone as a concomitant medication. There's the rationale for that is that we really have a pretty limited understanding of the effect of growth hormone on muscle, and whenever you're doing a clinical study, you want to eliminate as much as possible those factors that could be confounding. So that is why in any clinical study there will be as much as possible medical treatment stays stable, and other concomitant things that are not absolutely medically necessary are limited. So as to try and get the clearest answer at the end of the study as to the effect of the intervention.
Okay, thank you. And so, so I think we talked about the 80 mg dose already, how you made that decision to use that. I have a question. What about, as I was listening to all the outcome measures you're using, first of all, so you're doing time to stand twice. They have to do it once independently, and then they do it again as part of the North Star?
That is correct.
So, actually, though, generally, correct me if I'm wrong, Angela, but I don't think it would be done twice.
Usually you do the North Star and you just extract the data.
Yeah, so
but you said specifically that it was done. You said it was done twice.
In the North Star, you're grading. One is grading the quality of the stand, and then in the time to stand is the measured time.
Okay. So they have to do it twice. Once to be timed, and then once to be measured on quality.
Probably best to defer the answer to that question because I.
Okay. Yeah.
That's really. I actually had checked on this, and the response I received is yes, it would be done 2 times.
Okay. It takes 10 seconds, so about that. And what about wearables? Are you guys thinking about doing anything with wearables in your study?
Wearables? We won't be using wearables in the Racer 53 study.
Okay. And in terms of, this is, just looking forward, a sort of short-term time frame and then a longer-term time frame. But so if we look at and we think about you recruiting, and then there's a lot of other studies going on, especially, are you concerned, especially gene therapy studies, are you concerned about competition, if we label it that way, with some of the gene therapy studies?
I can take this one. We're not concerned of competition for gene therapy because Viltolarsen is an investigational drug with a specific population that it can help in the DMD community, and those are patients that are amenable to exon 53 skipping. While we're hopeful to bring this investigational drug to the DMD population, it will not treat all patients with DMD. Therefore, we welcome any other therapy that can potentially help our patients. So,
yeah,
yes.
Okay. And what plans do you have? I mean, assuming this is all successful, do you plan to pursue other exons?
Yes. Nippon Shinyaku is under discussion for other exon skipping agents. We have different programs in our pipeline right now.
Mm-hmm. So you are planning on pursuing some other exons. That's great. And we have another question about a requirement on dosing, and it's asking if there's a requirement that you dose up, higher than the 80 mg per kg that you're doing right now. But at this point, it seems like there's no other dosing requirements. Is that correct?
At this time, yes, that is correct.
Okay. All right. Well, I don't see any more questions. I want to thank both of you a lot for your time today. And I think we'll have to close the webinar.
Thank you,
Abby. Very much.
Thank you very much. Should any other questions come through afterwards, we'd be more than happy if you'd like to send them to us.
That's great. Yeah, we'll try to list some questions any questions that come in or we didn't answer, and we'll put them on the web. That'd be great.
Thank you. We appreciate that.
Okay. Thanks a lot.
Thank you.
Thank you, Dr. Clemens. Bye now.
Bye.